So the first thing that we're going to do is we're going to load a number of packages that we need. So when you first have run the basic script before you start this script, then these packages will already be loaded and you don't have to do this again. And now we're going to load the input data that we need for checking the effects of the cell cycle. So cell cycle analysis starts here in this step of the basic Seurat pipeline. So the step where we are going to normalize the cells using the normalize data function from the Seurat package. So we normalize the Seurat object and then we save the Seurat object that now contains both the raw and the normalized counts into an RDS file in the working directory on our computer. And this is the Seurat object that is used for the cell cycle analysis. So we're going to load this Seurat object. How are we going to load it? Well, we're going to load it using the read RDS function. So we have the data now, we have our counts. In the next step, we're going to load the known cell cycle markers. So I've explained, you're going to use genes for which you know that their expression is specific for one of the phases in the cell cycle. Where can we find these marker genes? Well, there is here a link in the script. And when you go to this website, he will automatically download an R data object file to your computer. So the list that we have downloaded in that way is a list of human genes, because gene symbols, gene names, these short gene names, are conserved between human, mouse, and rat. This list does not only work for human data, but also for mouse and rat data. If you work with another organism, then you can find a link in the script that will take you to this website, where you can find a list of marker genes for other organisms like fruit fly and zebrafish. So I'm going to load this list of marker genes using the load function here. So when I run this, you see that there are two vectors appearing here. So one with the names of the marker genes for the S phase and one with the names of the marker genes for the G2 and M phase of the cell cycle. As you can see, the gene names are in capitals. Okay. And that's an issue because in our Seurat object, the gene names are not written completely in capitals. Gene names are used as row names in the Seurat object. And you see here that the gene names all start with the capital, but all the remaining characters are lowercase. So if we want to match them to the names of the cell cycle phase marker genes, we will have to convert them into capitals. And because the cell cycle marker genes are human genes, so their names are written completely in capitals. And the data that we have is mouse data, so mouse gene names are written like this. So if we want to match these two, we have to convert the mouse gene names into capitals. Now, before I do this, I'm going to make a copy of my Seurat object so I can mess with the gene names. So in that copy of the Seurat object, I'm going to change the gene names. I'm going to convert them into capitals using the toUpper function. And you can see here that they have been converted completely into capitals. So now I can match them with the names of the cell cycle phase marker genes.